Dr. Kizzmekia Corbett earned her Bachelor of Science degree from the University of Maryland, Baltimore County, where she first developed an interest in viral immunology. She went on to pursue her master's and PhD in microbiology and immunology at the University of North Carolina at Chapel Hill. During her graduate studies, she worked in Sri Lanka, studying how human antibodies respond to dengue virus infection. Her dissertation focused on distinguishing immunogenic versus pathogenic antibody responses. Dr. Corbett is an assistant professor of immunology and infectious diseases at the Harvard T.H. Chan School of Public Health and the Schutzer Assistant Professor at the Harvard Radcliffe Institute. Her research focuses on how the immune system responds to viruses and how to design vaccines that trigger strong and lasting protection. She was a key scientist behind the development of Moderna COVID-19 vaccine, a breakthrough that has saved millions of lives worldwide. Outside the lab, Dr. Corbett is deeply passionate about mentoring and inspiring young people, especially women and minorities, to pursue STEM careers. Through programs like the Keystone Symposia Fellows Program, her Corbett Lab, and various outreach and mentoring initiatives, she works to make science more inclusive. She also serves on several boards, including Boston Mayor Michelle Wu's COVID-19 Advisory Board. In her free time, Dr. Corbett enjoys spending time with her husband and son, exploring Boston's performing arts scene, discovering new underground hip-hop, R&B, and jazz artists, and even tracking life's details in spreadsheets, often with a glass of wine in hand. What I most admire about Dr. Corbett is her dedication to public health and her ability to turn complex, cutting-edge science into real life-saving solutions, like her work on the Moderna COVID-19 vaccine. She represents the perfect balance of scientific brilliance and compassion for people, using her research to make a tangible difference in the world. I also deeply admire her commitment to mentoring others and inspiring the next generation of scientists, especially women and minorities in STEM. I identify with her drive to create change through hard work, perseverance, and a genuine desire to help others. Dr. Corbett's work connects immunology, molecular biology, and gene expression to understand how the immune system recognizes and fights viruses. By uncovering how our bodies respond at the molecular level, she's helping explain the biology behind vaccines and immune protection, paving the way for more effective and targeted vaccine designs. She co-authored the article titled Structural Serology of Polyclonal Antibody Responses. The main conclusion from this figure is that vaccination with either mRNA-1273 or MVX-COVID-2373 induces a diverse polyclonal antibody response that includes strong targeting of the N-terminal domain, supersite, but those NTD-directed antibodies are more vulnerable to mutations in variant spikes. This suggests that vaccine design might benefit from immunomasking or reducing induction of overly variant-sensitive NTD-specific responses. Panel A shows a composite 3D map of spike Fab complexes showing where polyclonal antibody fragments bind the spike. For example, many Fabs cluster around the NTD supersite and the receptor binding domain. Panel B shows epitope plots indicating frequencies of binding to various regions across the polyclonal response. It quantifies how much of the PAB population binds to each region. It shows that NTD supersite antibodies are a dominant portion of the responses in these animals. Panels C and D show composite maps and epitope plots of variant spike Fab complexes. They show how binding changes when using variant spike proteins. They often reveal decreased binding to the NTD supersite in variant spikes, while RBD binding antibodies may show more resilience. This is an article she more recently co-authored, furthering her next steps in vaccine research. Her work represents the perfect blend of scientific excellence and social impact. From leading the development of the Moderna COVID-19 vaccine to mentoring the next generation of scientists, she continues to shape the future of immunology and public health. Her dedication reminds us that science is not just about discovery, it's about using knowledge to serve and protect communities. She's an inspiring example of how passion and purpose can truly change the world.